Hello friends, welcome to my channel. This video is for class 10 students preparing for board exams. Today we are going to start with chapter number 1, Heredity and Evolution. This chapter is divided into two parts. The first one is heredity and the second one is evolution. So today in this part 1 we will first discuss about the heredity. But before starting with the chapter, please like and subscribe my channel for further videos. So let's revise the previous which you have studied. Which components of the cellular nucleus of living organisms carries hereditary characters? So the answer for this question is its chromosome. Next, what do we call to the process of transfer of physical and mental characters from parents to the progeny? एक जनरेशन से जो अगले जनरेशन में कैरेक्टर ट्रांसफ़र होते हैं उसको हम क्या कहेंगे? So the answer is heredity. Which are the components of DNA molecule? This you already have studied in 9th standard. So there are three components of DNA molecule. The first one is nitrogen bases. There are four types of nitrogen bases: adenine, guanine, thymine and cytosine. The second component is pentose sugar, in which deoxyribose sugar is present, and the third component is phosphoric acid. Let's start with the definition of heredity. Heredity is the transfer of biological characters from one generation to another via genes. कोई भी कैरेक्टर जब एक जनरेशन से अगले जनरेशन में ट्रांसफ़र होता है थ्रू जीन्स, उसको हम कहेंगे हेरिडिटी. So here are the names of the scientists and their contribution, which are very important according to the board exam point of view. This comes for match the following as well as in correlation or fill in the blanks. So the first scientist, Gregor John Mendel, he is the pioneer of modern genetics. Second scientist, Hugo de Vries, he proposed the mutational theory. The Walter and Sutton, they observed the paired chromosome in the cells of grasshopper. Oswald Avery, Maclean McCarthy and Colin MacLeod, they proved that except viruses, all living organisms have DNA as genetic material. Francis Jacob and Jack Monod, they proposed a model for protein synthesis with the help of DNA in bacterial cells. So the next question is, sketch and explain the structure of DNA and various types of RNA. So this you already studied in 9th standard. So here we will discuss it in very short. DNA, it is double stranded. It is made up of two polynucleotide chains that runs anti-parallel to each other. It consists of nucleotides, and there are four nucleotides that pair complementary to each other. That is A pair with T and G pair with C. Now there are three types of RNAs: messenger RNA, ribosomal RNA and transfer RNA. Messenger RNA क्या करता है? It transfer the character from genetic information from DNA to ribosomes for protein synthesis. Whereas RRNA अमाइनो एसिड्स को जोड़ने का काम करता है जिससे वो प्रोटीन बना सके. एंड द थर्ड वन tRNA इज यूज्ड ड्यूरिंग ट्रांसलेशन एंड इट हेल्प इन ट्रांसपोर्ट ऑफ specific amino acids to the polypeptide chains. Next, explain the meaning of genetic disorder and give the names of some disorder. So the answer for this is, a genetic disorder is caused by abnormalities in the genome and it can be caused by a mutation also. This means ऐसे disorders जो जीन्स में एबनॉर्मलिटी होता है, फॉर एग्जांपल अगर क्रोमोसोम कम या ज़्यादा हो गए तो उससे जो डिसऑर्डर होता है उसको कहेंगे जेनेटिक डिसऑर्डर, या फिर अगर वो म्यूटेशन से हो रहे हैं तो भी उसको कहेंगे हम जेनेटिक डिसऑर्डर. देयर आर सम एग्जांपल्स गिवन हियर: डाउन सिंड्रोम, phenylketonuria, Klinefelter syndrome, etc. Now, location of the genes. So, information about protein synthesis is stored in DNA. This sentence is very, very important again. Let's see this diagram. Each and every living organism is made up of cell, and each living cell consists of nucleus, and nucleus consists of chromosome. और जब हम क्रोमोसोम को और डिटेल देखेंगे, द क्रोमोसोम इज मेड अप ऑफ डीएनए एंड द स्मॉल पीस ऑफ डीएनए इज कॉल्ड एज जीन. नाउ सेंट्रल डॉगमा. व्हाट इज सेंट्रल डॉगमा? When DNA is converted to RNA and RNA is converted to proteins. So the definition for central dogma is the proteins are synthesized by DNA through the RNA. This is called as central dogma. So here the diagram is given of central dogma. DNA से first step में हम क्या बना रहे हैं? RNA. And this first step is called as transcription, in which we are making DNA to RNA, mRNA. And in second step, mRNA is converted into proteins. So this step, second step is called translation. So let's discuss transcription in detail. The process of RNA synthesis is called as transcription. And during transcription, the double-stranded DNA is converted into single-stranded form, and only one of the two strands is used for transcription. The sequence of nucleotide in mRNA is always complementary to the DNA strand used for the synthesis. तो ये जो mRNA बन रहा है उसके जो भी न्यूक्लियोटाइड का सीक्वेंस है वो उसके DNA के सीक्वेंस के कॉम्प्लीमेंटरी होगा. देयर इज यूरासिल इन RNA इंस्टेड ऑफ थायमिन ऑफ DNA. देयर इज वन डिफरेंस इन DNA एंड RNA, दैट यूरासिल न्यूक्लियोटाइड इज प्रेजेंट इन RNA and thymine is present in DNA. For transcription, the enzyme which is very important is RNA polymerase, that is used in transcription process. The mRNA formed in nucleus comes in cytoplasm. So ये process कहाँ पे हो रही है? सेल के न्यूक्लियस में. It brings the coded message from the DNA. और mRNA पे आफ्टर ट्रांसक्रिप्शन जो भी मैसेज आया है कोड फॉर्म में आया है, and then this mRNA will be transported out into the cytoplasm for translation. So the mRNA जो साइटोप्लाज्म में ट्रांसपोर्ट हुआ है, it consists of the triplet codon. Now what is triplet codon? The code for one amino acid consists of 3 nucleotides. It is called as triplet codon. Dr. Hargobind Khurana, he discovered the triplet codons for 20 amino acids. He has been awarded with Nobel Prize in 1968. So each mRNA is made up of thousands of triplet codons. और यही mRNA कहाँ पे ट्रांसपोर्ट हो गया है? It is transported to the cytoplasm for translation. So let's see the translation and translocation. As per the message on mRNA, amino acids are supplied by the tRNA. The tRNA has anticodon having complementary sequence to the codon on mRNA. This is called as translation. अब translation process में देखिए, ये जो blue color का दिख रहा है आपको strand, it is mRNA और उस पे है codons. 3 न्यूक्लियोटाइड्स का जो ग्रुप दिख रहा है वो आपको वो है कोडॉन्स. एंड टू रीड दिस कोडॉन देयर इज एंटीकोडॉन प्रेजेंट ऑन टीआरएनए. जो येलो कलर का आपको क्लोवर लीफ शेप दिख रहा है वो है टीआरएनए. और टीआरएनए के पास क्या है? एंटीकोडॉन. सो अकॉर्डिंग टू कोडॉन एंटीकोडॉन, टीआरएनए विल ऐड स्पेसिफिक अमाइनो acid. The amino acids brought in by tRNA are bonded together by peptide bonds with the help of rRNA. So जो ये राइबोसोम है, पिक्चर में आपको दिख रहा है राइबोसोम, ये क्या कर रहा है? अमाइनो एसिड्स को जॉइन करता है with the help of peptide bonds and forms the long amino acid chain. During this process, the ribosome keeps on moving from one end of mRNA to the other end by the distance of one triplet codon. This is called as translocation. तो राइबोसोम हमेशा one end से next end तक move होता है by the space of one triplet codon. And this process will continue till the mRNA till the ribosome reaches the end of the mRNA. So this diagram shows the mRNA coming out from the nucleus, and the second diagram shows the translation process in which ribosome is adding amino acids and tRNA is carrying the amino acid towards the codon. The next topic is mutation. Mutation is a sudden change in DNA sequence. Some mutations may be minor, but some may be major or considerable. This mutation may cause the genetic disorders like sickle cell anemia, and there are many other diseases which are caused by the mutation. So the important questions from this topic is given here. So you can review and check how many questions you can answer. And these questions are very important according to the board exam